talk about sort of the basic biology of a cochlea, which is sort of the basic structure to which we transduce pressure waves to sort of neural electrical signals. Now, I want to kind of do a biological overview of some of the important aspects that might be important, whether you're sort of just getting introduced to this sort of space of sort of cochlea, mammalian cochlea, human cochlea, or if you're actually looking at the computing aspects or interested in sound processing or speech processing. It becomes really important to get a sense of the various pieces and parts because many people will talk about some of these options and some of these operations, and so it's just kind of good to get a sense of where things are. So the first thing, you sort of look at the main part. And before I go any further, I will have to just outright admit I did not draw these pictures. Most of the time I draw almost everything I do. Some of this is just stuff that, just amazing pictures. I can't actually remember where I actually even got these. So I just want to make that statement and thank whoever did, and I happily give more credit and comments going forward. So what you end up finding is that inside the ear, so you have two of them, in the human case, your cochlea actually sits in the middle. It's a main sort of transduction element. You certainly have pressure waves that come through the ear. It hits the membrane, tympanic membrane, and there's a whole sort of bone structure in here that then guides that pressure wave into the cochlear structure, which is the sort of starting thing with all of this rolling in it, is where kind of the cochlear structure looks like. Now there's a couple other neat things to kind of anchor and understand what's going on here. One is there's a whole auditory nerve that goes to cochlear nucleus and related structures in other mammalian cases. So these are going to be sort of the output structures of what is happening out of this cochlea. You'll also notice that you get these semicircular canals, which are basically used heavily for getting balance right, effectively sort of position and velocity kinds of sensors. We see this a lot in other synthetic systems these days. So these are very, very interesting in terms of all the pieces there. Obviously there's some interesting spots that go into other parts, into other human cavities within the facial structure. If you have a cold, you use station tubes or something right here, this might be getting blocked up. Often becomes very uncomfortable. Also because it's blocked up will also mess up your sense of balance sometimes. So that's where everything is anchored to. That's kind of the basic structure. And I imagine there's many, many things we could talk about all of these pieces. But we'd really like to get to what's in the cochlea itself. And so this is a sort of rolled up structure. But if I were to take this structure and sort of roll it all the way out, what I would see is this sort of structure where I have here sort of the base structure, stapes, that's actually pushing fluid into this, right? So that's this little structure right here. And what I would notice is the structure tends to be a set of three different sort of fluid areas. One on the top, there's a hole on the one side here. Fluid then kind of comes back down the back side, and there's a window here. So as this pushes in, this window pushes out. Because basically fluids such as water and saline are incompressible. So if I push in, I've got to have a place for it to push out and vice versa. So we keep everything equilibrated. And then there's another sort of space in the middle. So what's interesting is that you get these pressure waves coming through over the cochlear structure, and then you have this sort of structure in the base, which is basically there's sort of a basilar membrane, which is doing most of the transduction. And one of the things we notice is that the size of the basilar membrane changes rather significantly as I go from the front side to the back side. And one thing that one will see going forward is that that has actually a major issue on frequency of where things might be more responsive. And that turns out to be very important for the overall structure of this. But the main things to notice is that you have an input here, you have pressure on the fluid, you've got a hole in the middle to which fluid comes through, and then fluid comes, and then it pushes back. So you might say, well, what does it look like if I took a cross section through here that would include the basilar membrane? Well, this is what that cross section looks like, and when you look at the edges, it's a very, very strong structure around the edge. Okay, and then you actually can see very clearly that you have three main windows, one of which is the top one, which is where the water and saline gets pushed forward, or pushed back. This is the primary element, but then it comes back through the bottom part of the membrane. And what happens is you have two different membranes. You have one membrane on the top, and then you have the other membrane down here, which is the basilar membrane. And this turns out to be one of the more critical structures of the whole thing. All of this is really important, and all the structure is there. What's going to happen is you're going to have this pressure wave that's going to sort of uniformly push down on this particular membrane, which is then going to influence what goes on in here. Some of that back pressure is also going to affect the structure on the bottom side of this. And so there's multiple places for coupling here. But most of the things that we're going to look at is that as a result of things moving, that this basilar membrane, this area in the middle, which is kind of being anchored and kind of has freedom to move back and forth, freedom to shift, is actually going to be going to have different kinds of vibrations on that part of the membrane. And what's going to happen is that this structure in the middle, which you can look at much closer here, you can see the basilar membrane. You can see that there's a number of other sort of things that anchor this. And what you find is you have a number of different structures here that are basically sensing the movement of this membrane. And so what you find is you've got a whole sense of outer hair cells and one inner hair cell. And the inner hair cell is actually effectively a transduction mechanism to say, well, how much is this membrane moving due to the change? And as a result, I'm going to then transduce that movement of that hair cell, that sort of hair fiber that's on that membrane, and that's going to then create an electrical signal that can then be transduced with neural signals. There's three other outer hair cells that sit over here, and these actually provide sort of active feedback on this membrane and on the overall structure to give you some form of local gain control over the structure. So we'll often talk about what's going on with the basilar membrane, its frequency dependence, and how you end up transducing that. Realize there's a lot of pretty amazing machinery all the way through here, and I really, you really could spend quite a bit of time, and there's been many people who dedicated their careers to look at these questions. So there's a tremendous amount of material all the way through, but it's important to kind of get a starting point to kind of know how to make sense of this, whether you're looking at this from a biological perspective, or if you're looking at this for wanting to understand the acoustics and acoustic science.